Are you here?
Hello. Yes, I'm here.
Yes,
Can you hear me?
yes, we
Thank
can
you,
hear
Basma.
you clearly. Okay, thank you.
Okay, hello everyone. Let's begin our presentation today about the basics of molecular biology in pediatric hematology. I have no disclosures regarding this lecture. Please, everyone, can you mute your mic so we don't have any interruptions? Thank you, everyone. Okay. Our outline will be an introduction, a type of mutations, techniques for this mutation detection, and application of techniques in hemato-oncology. I just want to remind you that a gene is a section of our DNA, it's our genetic code. It's like the blueprint of our body. If you think of the DNA would be, and the genetic code, it's like the Lego blocks that build up the proteins in our body and cause or our phenotypic changes and appearances. The genetic mutations is the change in the DNA sequence, as you can see, the change in any of the assembly of the 4 nucleotides that we have, the adenine, cytosine, guanine, and thymine. The genetic mutations actually are the drivers for oncogenesis, so that we have a mutation that is caused in one cell, and this cell gets clonally amplified and gives us the tumor that we have in the hematological malignancies. This clonal malignant cell, according to the mutation that happened in the stem cell, the lineage specific cell, so it's either myeloid or lymphatic, and the myeloid or lymphatic according to the commitment of the cell, either in the myeloid series or the lymphatic series. So what causes the mutations? It's either in the replication or exposure to mutagens, radioactive substance, X-ray, radiations, carcinogenic chemicals, viral infection. We all know that there is a huge role in viral oncogenesis. So whatever the cause that implicates the mutation, it leads to the carcinogenic event and the clonal growth of the malignant cell. Our this type of mutation is the point mutation. As you can see here, it's a substitution mutation where the T is replaced by the C. It's just as if think of it as the type of mutation or the spelling mistake that we have in the sequence of the DNA. And mutations, point mutations can be a substitution mutation. As we can see, the C is replaced by the T, or an added mutation, added nucleotide or deleted nucleotide. All these count as point mutations, as mutation in one base only. Insertion deletions also are part of this point mutation type or classification. And there are the chromosomal mutations. They take an entire section of the chromosome. It might be deleted, duplicated, and as Dr. showed us in the cytogenetics, it is a structural mutation, takes a large amount of DNA. So as you can see in the right figure, we can have either a deletion in the chromosome, a duplication, an inversion, as you can see here, an insertion of part of the chromosome in another chromosome, or a balanced or unbalanced translocation, where part of one chromosome goes to another part of chromosome, causing us what's called a fusion gene, where we have you know, the arrow, this is the fusion gene when one part of chromosome with another part of chromosome. And this, and you see on the right side figure, you can, this substitution, insertions or deletion cause a change in the messenger RNA and consequently a change in the amino acid sequence, and this causes a change in the protein that is coded by the DNA, and this change in the protein can either lead to a tumor suppressor gene being deleted or suppressed, we have a mutational event, or anything that causes and increase tyrosine kinase activity. So whenever we look at the hematological malignancies, we have to have an integrated diagnostic approach where we can look at the phenotypic results, that's the morphological assessment of the blood and bone marrow, the flow cytometric results of the cells and their commitment. And then we have to look at the mutations itself that causes this phenotypic aberration, the cytogenetic abnormalities, if it's a larger structural aberration, or whole genomic sequencing, or a larger, as they say, a holistic bird view eye of all the mutations or the events that happens in our malignant clone. So I have a question for you, everyone. What is the aim of molecular mutation detection? What is our aim? If you have to scan the code, please, and give me your answer. Why would we do molecular testing? You can scan the code and type in your answers. What is the aim of molecular testing, in your opinion? If you want, I can put, can you see the code now? I can put a link.
Dr. Sally, the code is
Yes.
very small. Can you make it bigger? The code is very small. Can you make it bigger? Or if you can put the link in the chat box?
Okay, I will try my put the link in the chat box. Maybe easier.
Thank you, Doctor.
Okay. Okay, everyone. Yes. Okay. Actually, you all got the answer right. Risk stratification, targeted therapy, diagnostic, uh, prognosis, targeted therapy, treatment, monitoring. I can see all the answer right, and they all have the rationale behind the molecular testing in pediatric hematology. But in my point of view, if you would want to find the aim behind the molecular tests that we have two approaches to think of them. Those molecular tests, are they qualitative versus quantitative? If you go to the other slides where you answered, you all said risk stratification, targeted therapy, prognosis. All these are qualitative diagnostic techniques or molecular diagnostic techniques, where we have for the classification criteria, for the prognosis, identification of molecular targets, versus the quantitative molecular techniques, which they monitor. We have a specific genetic alteration that we monitor over time for follow-up, and we have the minimal residual or measurable residual disease assessment. And if we think of it like that, in this way, quantitative versus qualitative molecular tests, then we can assign the techniques for ordering these molecular tests. Either qualitative techniques, so the qualitative techniques give us a yes or no answer. If a mutation is found or not, and what is the mutation. So it doesn't give us any quantity-like information. So these qualitative techniques are either polymerase chain reaction-based techniques with all its modification, either restriction, fragment length, polymorphism, allele-specific PCR, and sequencing, Sanger sequencing techniques, and capillary electrophoresis or fragment analysis techniques. All these techniques give us qualitative results. Versus the quantitative techniques that gives us amount. Whenever there is an amount, then there is follow-up, then there is decrease and increase, and there is measurement of measurable residual disease. Then the techniques at hand are the quantitative PCR, the digital PCR, and the next generation sequencing. So to summarize all the molecular techniques, they all go through these basic steps. We have to extract our nucleic acid, either DNA or RNA, from our sample. For the DNA, we go for amplification directly. For RNA, we have to add a reverse transcription step. Then with the amplification, we have all the categories that we said for the conventional PCR, the ASO PCR, the quantitative PCR, the nested PCR. And then after the amplification, we have a lot of other modalities. We can go either for Sanger sequencing, we go for high resolution melting curve analysis for differentiation of different alleles. We have electrophoresis visualization either by gel or by capillary electrophoresis in the fragment analysis. So to explain what is the PCR or polymerase chain reaction, we have our primer sequence, as you can see the orange here. We add them to our template DNA in the presence of the DNA polymerase, and these flanking primers, as they say, they flank the area of sequence, of target of sequence that we want to amplify, and in a many cycle of denaturation annealing, this copy of DNA is amplified millions of times. So if I take what was called a sequence-specific primer, so I will add the primers that are for the targeted sequence that I already know, and they will amplify the sequence of the mutant type and the sequence of the wild type, and I can detect this mutant or wild type via gel electrophoresis or via, even if these labels are primered, are labeled with a fluorescence, we can detect them by fluorescent labeled qPCR. As you can see here, for the fusion genes that can be detected by cytogenetically, the translocations, for the molecular, we call them fusion genes because we work on their molecular level. For the cytogenetics assessment, they are called translocations. They are synonyms. They represent the same target. However, we look for a gene by molecular. For the AML, we have the AML1 ETO, that's RANX1, the PML RARA, core binding factor, the FIP1 platelet derived growth factor is for myeloid. We have in the middle the BCR-ABL that's found in the myeloid and the lymphatic, the CellTal for the TLL, MLL rearranged for BLL, E2APBX, and TEL-ML1. Their names are the 119 translocation and the translocation 1221. So all these fusion genes can be detected via PCR. Their absence or presence. I'm talking now in the qualitative setting. As for the Sanger sequencing, we have the D-dioxynucleotides that are labeled, fluorescently labeled, ATGNC. We have, they terminate the chain of sequence, the sequencing amplification. They produce different fragments, and where they are run on the capillary electrophoresis, we have different fragments that translate into the sequence that we want. Okay, it's the same. You can see that the terminated fragments are all labeled, and they are they run on the electrophoresis solution. So we have the sequence is aligned. And some of the mutations that are detected by sequencing, because they are large, the conventional PCR can detect point mutations perfectly. As we know the point mutations that we have, exactly, we can design primers specific for this point mutation, and then we can use these primers for PCR. However, whenever there is a larger sequence of interest, not a small gene that we can amplify, or multiple targets of the in the gene in multiple exons, then we have to have a larger segment to be sequenced, like the JAK2 exon 12 mutations in myeloproliferative disorders, the MPL, the calreticulin, and SEPA in AML. As you can see in the 2022 WHO classification of AML, it's all based on either fusions, genetic fusions, rearrangements, or mutations that all can be detected for diagnosis via the qualitative molecular techniques that we said before. And another application, as we want to highlight, is the BCR-ABL kinase domain mutations. These mutations offer resistance to first-line TKIs or tyrosine kinase inhibitors, so their detection would be via the Sanger sequence. They are, as you see here, point mutations that target the choice of treatment. And we can see here in the simplified version, these mutations with their corresponding line of treatment, either first, second, or third, or transplantation for the T315I mutation. I have another question for you, if you all with me. Sorry. Can you see the slide?
Yes, doctor, we can see
it. Can you see the question? If I want, I can put the link also in the... Can you see now the question?
It's very small.
Yes,
okay. Okay, just a second. You have it now in the chat box. You can. I can read you the question. The boy is 13 years old. He presented with DIC, increased promyelocytes in the bone marrow aspirate. What do you think the test that should be ordered, and which technique? Okay. Okay, PCR, translocation, PCR, PML rara, PML rara by fish, PML rara molecular, PCR for PML rara, PML rara by PCR, PCR. Okay, come on, everyone. Okay, we have only. Okay, all of you are right. Someone said translocation 1517 by fish. Fish is okay for the diagnosis, but and it has some sensitivity also. But for follow-up of the disease and the measurable residual disease, you would want to start your ordering with PML rara by PCR, by qPCR, that is quantitative. So you can have an initial knowledge of the burden of the disease so that you can follow up and know your treatment response is going which way. If you are giving the treatment Pediatric, then you would want to monitor if it's working or not. How would you monitor if you're not using a quantitative technique that can measure the quantity of the target insult, that is the 1517 translocation or the PML rara fusion gene. That gives us the advantages of the quantitative techniques. The first quantitative technique that we have is the quantitative PCR. It goes around amplification of a target mutation versus normalization with the housekeeping genes that we know, so that we know that this sample, we normalize the target versus the housekeeping gene of this sample, and we run it. This target is fluorescently labeled, so that we run it on our qPCR machine. We have, as you can see, these curves where they cut off, they cut the threshold, and we say this cycle threshold of the amplification, and we can quantitate these points, as you can see, and translate them into quantities of known number versus standard curve that is run with the sample with known quantities, so that we can infer the slope of the standard curve, the amount of target, the fluorescency target that we have in the quantitative PCR setting. In this setting, we have to know for sure what the target that we are looking for. Like the case we have the PML rara fusion gene. That target we already know, and we have designed primers and probes for them, fluorescently labeled. For all the fusion genes, we have this setting for the quantitative PCR for the targets. For the point mutations, or other mutations in the nucleophosmin gene, we know that. So we can have a quantitative PCR design for this target itself. The second technique, which is quite newer than the quantitative PCR at least, is the digital PCR. It's not that new, but it's just newer than the quantitative PCR, where we use the principle of the quantitative PCR, except in another setting with the idea of partitioning the PCR reaction that we saw earlier, the amplification and denaturation and extension. We separate this in different partitions so that each partition in itself is a PCR reaction. So instead of having the collective, as here, this is the collective of all the molecules that are there in the solution in the quantitative PCR, we partition the solution in hundreds of thousands of partitions so that the machine can detect a single molecule. And whenever the machine can detect a single molecule, this gives us the power of absolute quantitation. So if we can detect the single molecule that is positive for the mutation or the negative for the mutation, whenever we add them together, we know the number of molecules that are there as an absolute quantitation without the use of normalization, of standard curve inference for the cycle threshold or the CT. And this gives us another added value of detecting targets or mutations found in very low abundance in the measurable residual disease, where we look for one cell in 10,000 or 100 or million wild type cells. So as I was telling you, the qPCR versus the digital PCR, it gives us, as you can see, the digital PCR gives us dots for each molecule amplification, and we can detect molecule by molecule which molecule has the target and which one doesn't, and we can add them in an absolute quantitative way versus the quantitative PCR. As you can see, it looks at many molecules versus wild type. They go for the collective fluorescence from all molecules together. And it gives us relative quantification. So for the quantitative techniques, we would use them for, as we said, treatment response follow-up and measurable residual disease. And for the response monitoring, the famous qPCR and the log reduction in the molecular response in the BCR-ABL1 in the chronic myeloid leukemia. We all know that the transcripts that are there, we measure the transcripts via quantitative PCR, and we have this reduction in whenever we go for the deep molecular response, we have this reduction in the transcript or the target of BCR-ABL1. As we go for the next generation sequencing, this is the third technique that can quantitate the target mutations. It differs from the Sanger sequencing is that the Sanger sequencing sequences only one molecule at hand, only one DNA fragment. However, the NGS can amplify and sequence more than one target. And when I say target, I mean gene or mutation simultaneously per run with millions of fragments detected. So it allows for multiple target detection and quantification in relation to each other and in relation to a housekeeping gene or a normalized gene. In short, we have this DNA library sample. We have the genomic DNA. We go for either amplicon sequencing or targeted sequencing. We add our adapters and ligation, and we have the sequencing machine that goes for all these targets to sequence them simultaneously per run. As you can see, for the AML, we can target all these genes in one go, and we have their amount also in amount of reads per sample. So for measurable residual disease, it detects levels of residual leukemic cells that are persisting. It tells us about the outcome prediction of in AML and in ALL. The techniques that are used is the quantitative PCR. Also, it is used for measurable residual disease because it can quantitate the NGS. For MRD, for AML, because it can target many targets at once. And the multi-parametric flow cytometry, which you will learn in the next or the next lecture or after it, I think. For the AML, for the myeloid series, we have the nucleophosmine as target. We can use it. We can use the method either quantitative PCR, digital PCR, or NGS, either on peripheral blood or bone marrow. And whenever we start with the peripheral blood or bone marrow, we have to continue with the same initial sample that we have taken. If we begin with the peripheral blood, we can go for the peripheral blood in the follow-up. If we begin for the bone marrow, or we can follow up with the bone marrow. However, we have to stick with the initial sample that we have taken to make a baseline assessment of the clone and the target that we have. We have core binding factor AML, the inversion 16 and the 821, ranixtronics, and the APL, the acute promyelocytic leukemia, translocation 1517, also by qPCR and digital PCR. This is for the myeloid in the measurable residual disease assessment. How about for the lymphoid malignancies? For the lymphoid malignancies, we have the VDG rearrangement of the B and T cells, or the B and B cell receptors of the antibodies and the T cell receptors. They have this unique sequences of their B and T cell receptors that are caused by the joining of the V region and the D region and the joining region to give us this unique sequence that is a genetic sequence of the clonal event in the lymphoid series that has clonally increased. So for measurable residual disease monitoring of lymphoid malignancies, we go for the TCR and BCR rearrangements, the VDG rearrangements, also via NGS. I will end with the whole genome sequencing. This is a qualitative method. It's not quantitative. However, it gives us an overview of all the genes throughout the whole genome without prior knowing of the targets at hand. So we amplify the whole genome of the sample or the patient. We cut them. We have our library. Then we sequence the whole genome, and we align the genome that we have sequenced to a reference genome to know which variants are there in our sample. And when you get the whole genome sequencing report, you would find variants that are found in the DNA. They are the primary findings that are there. They are labeled either pathogenic, that this means that they are disease causing, as known from their sequence, from their function. If it's loss of function, it's a stop codon that causes the protein truncation, and also the presence of absence of this mutation in databases that they prove that they are disease causing. Or you can be likely pathogenic, or variant of uncertain or undetermined significance, where we know that there might be a loss of function. However, low studies on this, not many studies have reported these mutation in the database, of this variation in the databases. So we don't know, we don't have any sure knowledge of their effect or disease causing effect. And we have the likely benign or benign variants. These variants that they don't cause any change in the protein sequence. They are not reported to cause any diseases. So they are just variations in the sequence that happen normally within individuals. As you can see, a lot of work has been going on through from not 2 years ago about the use of whole genome as a sole diagnostic method to detect all the aberrations in BALL. As we can see, it can detect all known and unknown mutations. So in comparison to the targeted individualized approach, either sequencing, the whole genome sequences tells us everything on in the whole genetic material of the sample and the patient. And it has been there as a routine diagnostic tool in all children in the NHS. And they also found that it gives us a bird's eye view about all the implicated targets in the whole genome, so we can use any of them as a target for follow-up, and we can see the relation between them and each other, and we can use all this data also for treatment monitoring and for targeted treatment. So I finally finalize my presentation. I just want to ensure that you know that hematological malignancies arise from clonal carcinogenic mutations. Mutation detection techniques are guided by clinical decision. So whether you want to risk stratify or follow up or detect MRD, you have to choose the technique that gives you the answer. Quantitative mutation detections, qualitative include the PCR-based technique, Sanger sequencing and next generation sequencing. The quantitative measures or techniques are the qPCR, digital PCR, next generation sequencing, and the whole exome sequencing offer better approach than individualized mutational detection techniques and targeted panels. And thank you.